biodiversity, maybe we need a quicker method, a shorter method of actually finding these. Would primers do? Would significant primers, which are specific for each of these species, be enough? I don't know. I'm not a biologist. I've always been working with the computer since 1977. So I'm not really the person who should be answering this question. I would like to refer it to some of the other people who can come out with a rapid way of estimating the biodiversity in a particular area. And therefore, we have a way or a means to track loss of species. So that is be my concern. And while it is very noble to do all these things, are we actually addressing the problem particularly? You know, when I first started BSc in 73, I hate to tell you I'm really old, but the first lecture I attended in science was precision and accuracy. We can be very precise, but are we being accurate? Are we actually focusing on the problem at hand? That would be my call. My worry is that we have lovely methods of machine learning, and we have excellent methods of actually storing data and retrieving it and analyzing it. We always believe that we have sufficient data to do data mining data, and we can actually build models on them which will be predictive and within reasonable accuracy, and we have a plethora of measures to actually measure that accuracy. We also do machine learning. It used to be called something else in the old days, but now it's very popular, and if you don't do machine learning, you're not in the crowd, isn't it? So we're all doing machine learning, but how much machine learning is going to help? By the time we gather the data and do the machine learning, we already lost some of the species. So, what is it that we should be trying to do? And will photography help? They may identify large species, they may identify patterns of migration and movement, but will they actually be able to count the species in a particular area accurately and give us the health of the biodiversity in a particular region and how whether a particular region is unhealthy? What is the result of all this crop burning that we hear about? Are all the fumes going to affect the biodiversity? These are all issues that we should be looking at. We should be seeing how we can conserve the environment. After all, our Vedic tradition was to actually keep the environment pure. We are always talking about purifying the air, right? So we should actually be thinking how can we maintain the biodiversity in a slightly coherent manner, and how can we estimate it quickly and efficiently, while we would love to sequence every genome that is there is available, and in the process get publications and things. Is it feasible? And what is the depth to which we need to do it? Do we have a model organism or a reference genome on which we can quickly do the analysis? These are all issues we need to put in, think about before we put in the man hours. That's my honest opinion. I'm not against genome sequencing. I'm not against looking for SNPs, especially in disease families. I heard this wonderful talk by Dr. Vinod Skaria, which was really insightful. And I was thinking, here is a person of such stature that I would be very happy to meet and collaborate with. So we should actually be focusing and therefore getting our efforts in the area that's going to give us the maximum benefit towards the common goal. I think we need to actually think about this. We have wonderful methods, we have wonderfully trained people, but how can we do a shortcut? How can we quickly figure out what's happening? That's what we need. So, can we actually address that question? Here, the people who do the sequencing will be able to help, not me.
Yeah. Yeah, thank you very much, Professor Shobha. How about others, please? Professor P.K. Gupta, Girish, Dev, Abhi, or Pratish, do you have any points towards these shared pathways?
I have one point to add to Ma'am Shobha. She has rightly said, and first of all, we should thank her. She being so enthusiastic and very supportive for young researchers. That is very, very important, uh, uh, because one more important thing which I realize, there is a lot of data, genome data, and when we speak about genomics, just sequencing and putting a lot of data, there is a lot of noise in that data. Rather than, at least in the Indian context, what I understand, whatever sequencing projects till date, to my knowledge, I have never seen a chromosome level assembly of any organism. Almost all of them are draft assemblies. And we are in a hurry to publish, which we should not. Even I also accuse myself in that process, because that's how we are trained, but that's not the proper way when we are really approaching towards biodiversity research. Biodiversity research really, really hard, and not only hard work, it is a time. It requires a lot of time. In the sense, what I mean to say, it requires a lag phase of research. 10 to 15 years of silence research work without much noise, without much uh, Allah Bulla about this, and we should try to come out with real information from those genome data, rather than putting them in the public domain, and without curating those data, and in a rush to publish, we miss the real information belied in this genome. So which is, I think, a gross injustice to biodiversity conservation in the Indian context. So we should try to improve upon our skills in understanding the data that is already lying in the public domain, and also, if you are really interested to come up with new projects related to big data and genome sequencing. That's all I would like to point.
Yeah. Thank you, Dave. Wonderfully inserted. I mean, I apologize. That we are just running short of time. So, I mean, moving to the third point. I mean, which certainly coalesces with the first 2 points as well. I mean, the third point is more not really associated with the non-coding RNAs, which I really wanted to discuss. But there is something called the use of environmental RNA, which we called as eRNA. And at some point of time, environmental DNA was was well thought about, but the use of environmental RNA, for species identification, remains unexplored. If you really see, by and large, for various reasons. But there has been interim some lines of evidence saying that yes, we have some RNA that may be abundantly say excreted by the organisms, and probably if at all, if that excreted RNA, which we call it as environmental RNA, that is available in the environment, that could probably allow us to reconstruct some gene expression data, or maybe community based genomic data, or community composition, something on those lines. So what are your visible thoughts on these directions? To start with, probably Girish.
But Prashant, in addition to non-coding RNAs, should you
Yes,
not
sir.
look at the whole epigenome? Because
Very much, sir. Yeah.
because there is histone modifications, DNA methylation, and non-coding RNAs. So very little is being done in India in the area of epigenomes, and that is something I think needs attention.
No, very much, sir. I mean,
We are doing a lot of work on non-coding RNAs, but not very much on histone modifications and DNA methylation, in my opinion. And data should be collected at the whole genome level. Genome-wide histone modifications, genome-wide DNA methylation, because these two areas and the work which we have been doing, we found, particularly with respect to disease resistance, lot of role is played by DNA methylation and histone modifications. And this area is almost unexplored in India.
no, very much, sir. I mean, more precisely in plants, if you really look into this, there could be some unexplored regions more towards the open chromatin elements, sitting 5' UTR regions. Those particular regions might be wonderfully exploited. That could really help us disseminate understanding disease resistance in plants, for example. That was also my a subtle point, sir. I mean, the reason why I probably mentioned the non-coding RNAs is in more relevance to the environmental RNA that is really considered to preserve the community that is really thriving in the environment. So the eRNA is really playing a big promise in the near future. I think ever since that 2019 November article that really came up, where the authors have wonderfully described how the environmental RNA that is really made available from the organism's excreta or whatever, that could probably allow us to reconstruct the phylogeny or reconstruct the community composition or the gene expression data or whatever. I think I thought sir, maybe this is more relevant to plants. I think a large amount of work is really being done in these directions. So any valuable insights from any one of you would be nice, sir. Please.
Right.
Girish,
Right.
please. Sorry.
Rashi, if I can add,
Yeah.
actually, I want to highlight what Gupta sir just now said. The idea of understanding. See, I mean, there are some concerns. First, let me say I'm not a bioinformatician. I don't do the bioinformatics myself. By an end user of bioinformatics, I work with people like you, trying to understand the biology behind it. So one interesting point I want to make in the context of biodiversity and epigenome. I have been following this work, and some of that we are implementing. They just studied this epi-recombinant middle Arabidopsis and to see in the context of weeds how it performs. Okay, so when there is a diversity, epigenetic diversity is there among the Arabidopsis lines, their yield and the performance in response to pests, other weed plants, and environmental challenges improve. Okay, which is really not a many work. I mean, this is just published very recently in one year ago or so. Obviously, the shift is now, as sir pointed out, certainly that really surprised me. How you take one particular species, you alter the epigenetic landscape and create several lines among them, and now you put them together and put them individually. They perform better when they put them together in a combination than when they're alone. Okay, that really means it's published in a really good journal. We can, if you go by, if that data is true, it's really very encouraging for the plant, the crop people working on crop plants. And that's one of the reasons we propose to generate these lines in rice and tomato. And luckily DBT has approved our project, which we are working on it. If it is few years down the road, but when we eventually have such kind of lines, people can start exploring that. And if it can help, I'm sure, I think it would be a significant benefit in my understanding.
Yeah, wonderful, Girish. I mean, congratulations on that initiative as well. Yeah, thank you. So, Professor Shobha has a point, please. Yeah.
Yes, sir.
You were
Prash,
saying something.
this eRNA, eDNA may be a way to actually scan a lot of species in one shot from an environmental sample, and they have successfully done it for marine organisms. So Dr. Abhishek may be interested in that. So maybe water quality can be done with that and stuff like that. So this is the paper by Sato et al. Is also about water, where they found the schistosoma samples in China. So maybe that's something that is worth exploring and something that can give quick results. That might, while we are waiting for all the whole genomes to be sequenced, that might be a good opportunity.
Yeah, wonderful, wonderful. So, if there are no points on this, I also would like to add one of these particular points. I mean, we are talking about so-called helminth parasitism. I mean, which is quite phenomenal in northeastern India. We are at one point of time we are trying to eradicate these helminth parasitism from geographical niche region like northeastern India. But we should also be conscious that there is also helminth biodiversity that is really rigorously maintained in rats, where some countries like Estonia and South European countries, they're extensively working. While Professor Shobha was giving this particular talk, and I was just going through one of these particular literatures, and that's quite fascinating. I mean, that really begs us a question that we in India probably, thanks to wonderful researchers like Dave, who are really working in these particular directions. But there is a big lacuna where we are not probably progressing further. So my point is also more towards we are really sequencing these umpteen genomes. We are saying that yes, we are sequencing so many genomes. Now we have got 10,000 genome project, which is a 400 crore worth project from government of India. Now after that, what? Are we really succumbing to some kind of pressures, or are we really maintaining some kind of healthy repository so that we can get a rigorous data that is really captured for a better artificial intelligence. That was my particular point as well. So one final thing. So we have got about six panelists here. So each one of you would be given about 1 quick minute, please. What would be the foremost concern and challenge probably for you to ensure that a healthy biodiversity is maintained in India using the so-called the now generation sequencing data? 1 each minute, each one of you for the one final round, please. So starting with Professor P.K. Gupta, please. Yeah, please take 1 minute, sir. Please.
Maybe we can all. Maybe we can switch our video on.
Yeah. Yeah. Professor Shobha, you may just give your thoughts, please. In the meantime, sir comes. Yeah.
So I seem to be talking a lot as always. So, but what I would like to say is that the some of the take-home messages from listening to everybody is that we have some huge collection of expertise here. What we could ensure is firstly that all the data goes into a repository. It can be actually a government repository only available to the scientists in India. It doesn't have to go to NCBI. And maybe people like Dr. Preetish can help with organizing it, because he was talking about blockchain and access and things like that. And maybe we can actually get this going, because then we would know what we have and what more needs to be done. That would give us a very good estimate of how to proceed, whether it be plant or animal or helminth or human or whatever. So if we have everything in one place, maybe it will be a great resource for our scientists, who can then see the width and the breadth of the data and decide what will be worth studying using machine learning, or what would be worth doing more to collect data on, and how to actually analyze and get some benefit out of this data. So that might be something that I would like to suggest.
Thank you. Thank you. Thank you so much, ma'am. Yeah, Abhishek, do you have any say, please? Yeah.
Yeah. So
one quick minute, please. Huh?
Yeah. So basically, I agree with Dr. Soba that one needs to have a common platform where data is shared, and with worms, we have an issue to solve. And recently, people are trying. So people at different levels are trying. Suddenly, one day I got an email from an ICMR scientist to work on genomics of worms in relation to in a similar area, and we just participated in. So people once as a team, as a core bioinformatics team or genomics team who are pulling out the information, more people will be interested in working. Many people think that computational biology or genomics or bioinformatics are not their cup of tea, and they're scared of it. But I randomly found somebody dropped me an email. I don't know her, and our grant is successful. So basically, there's a lot of people who are trying, and they have laboratory resources or arrange the resources somehow. And we need to come up openly, diverse our information, which is able to reach to tier 2 cities, tier 3 cities, and even to villages, so that we can actually, we need a common platform where we can pull the data, and we have to keep data open so that somebody else is rapidly able to use it if they want to use it. So the one of the problem I see that there are genomics data, there are SARS-CoV data, but if the data is not fully openly given from Indian portal, I don't know whether I'm going to use it. And if it is not in portal, any portal at all, it's a massive loss in understanding any kind of biology. Biodiversity is even more challenging. Yeah. So yeah, with this I will end up. So we need a common platform and open-mindedness to work together.
Wonderful, Abhi. Yeah, I agree. Professor P.K. Gupta, please, sir. So what would be one single most point, one final bottom line?
You see, when you talk about biodiversity, our major concern is about microbes also. But there should be separate groups dealing with microbes, human health, and crop improvement, in my opinion. And among the microbes, I think we need to attend to the problem of microbes. Microbes, the pathogens. In most of the crop diseases, I know that lot more is being done on the crops, but not so much on the pathogens. So genomics of pathogens should be studied in great detail in order to find out what are those causes in the pathogen which cause the disease and how to handle that at the pathogen level. Because we are working on leaf rust for some time and we find that we know quite a bit about the genes for resistance among the crops, but we know very little about the pathogens. So there should be a group which should deal with pathogens, genomics, and other aspects of the pathogens which cause disease, both in case of plants and also in case of human beings, as Shobha has been talking about. A lot more needs to be done on the microbes which cause a large number of different diseases. That's all I think I want to say.
Yeah, thank you very much, sir. Very, very insightful. Dave? Yeah.
Thanks, Pras. I don't have much to say, but I will highlight certain points. Basically, for understanding the biota of a region or of a country, or let us say at the international level, the most important thing is that the species richness patterns. This is a major challenge. So the knowledge that we have, or the knowledge that has been accumulated over years, it is not sufficient. Every time things are changing, new species may be there. So species richness patterns and their accurate monitoring is a big challenge, which I think machine learning, which has been aptly discussed by our learned scholars here, would definitely help. But there has to be new methodologies, and there should be a coherence in biodiversity study patterns among all the research groups in the country. That is one most important thing. Apart from the scientific aspects that we have discussed, we need to understand the legal and the ethical issues concerned with the biodiversity. And since we are discussing here from Indian context, so we the researchers have to be seriously sensitized, and we should be all aware about the various legal aspects and the ethical issues with the biodiversity of our country. That is more important before we take up any particular biota, be it flora or fauna, for research purpose. That is what I would like to put at this point of time.
Yeah, wonderfully inserted, Dave. So since we are running out of time, I would like to summarize today's panel discussion. First of all, I would like to thank all the revered panelists here who have really given wonderful insights. We basically discussed four important points. Coalescing the need for the machine learning and artificial intelligence, given the plethora of genomes that are really sequenced to maintain the biodiversity. I think Professor Shobha, started with the role of plastics in the environment. That is really making a big havoc. And Professor P.K. Gupta, he emphasized the need for the genomes to phenomes. The phenome data is really missing, at least in terms of the plant phenotypes that we have. That is certainly a kind of a thing. Dr. Krish, he emphasized the need for certain sequence specific regions that could really allow us to understand the biodiversity measures. He has really taken an example of MGR5 gene. Dave has wonderfully exploited the blockchain technology, the need for ensuring that the time lapse that is entertain these given the lack of these so-called basic basic principles in maintaining the biodiversity. That is where we were discussing about the Earth Biogenome project and how yes, it is wonderfully sequenced as many marine genomes as it could, and we could get about 25 samples. That was what it was mentioning. There's also collaborative spirit as a strategy point. And Pritesh, of course, we were talking more the 10,000 human fully launched, and our points were more towards what is it after we sequence these 10,000 genome projects, could we really build some kind of strong principles and healthy measures in maintaining this healthy biodiversity. And of course there was also a point that Abhishek mentioned about the insect data, where the insect excreta could also be used for bringing up the RNA-based phylogenetics that we were really talking when talking about the environmental RNA. And we also spoke about the association mapping, which is actually a very important point that Pritish has raised. Association mapping plays a very important role. So it's not just sequencing these particular genomes, but a kind of a valuable time that can really look into these kind of things. And finally, Dev has given these wonderful insights about the biodiversity omic approaches that we were talking about. I think this has been a wonderful session. It's been a kind of a learning experience, taking all the things, the bacterial, eukaryotic, eukaryotic as well. Some of them were also talking about some of the insect and the animal and the pig data as well, and of course, the plant and the microbial data. I think we should thank all the panelists, with a big clap. I think there are no virtual claps, but we can also ensure that we can unmute our microphones and have a big thumping applause to all the panelists. Thank you so much. Thank you so much. So we will wait. We'll start our session in about 3 minutes, please. Yeah. Thank you so much. The next session will be chaired by Dr. Krishnamohan Medichella and Dr. Vinay Panda.